All right, guys, welcome back to Underground Science. In this video, we'll be talking about molecular mimicry. What we're going to do is we're actually going to jump in right into an example, okay, a real life example. We're going to be talking about Guillain-Barré syndrome, okay, and how this relates to molecular mimicry. All right. So let's say, right, you eat some undercooked poultry, some contaminated food with a certain bacteria called Campylobacter jejuni, right, or jejuni, Campylobacter jejuni. And so let's say you ingest this Campylobacter jejuni, right, and it goes into your stomach, then into your intestines, and makes its way down to the ileum, right? Let's say the ileum of the small intestine. And then once it makes it down to the ileum, so right here, right now, this Campylobacter jejuni can then enter your epithelial cells, right? Because remember, what type of pathogen-associated molecular patterns does Campylobacter jejuni have, right? It has flagellin, right? So it can activate toll-like receptor 5 on the cell surface of your epithelial cells or your enterocytes. It also has certain PAMPs, right? Like lipo-oligosaccharides, right? So LOS, which can activate toll-like receptor 4, right? And then it also has CpG DNA, right? Unmethylated CpG DNA, which can activate toll-like receptor 9, which is present on the endosomes, in your endosomes of your epithelial cells, for example, right? So, of your B cells, for example, sorry. So your B cells, your dendritic cells have can take in this Campylobacter jejuni and when they phagocytose it, right, they're going to form a phagosome, and inside the phagosome or the endosome, you have toll-like receptor 9 that can respond to and get activated by unmethylated CpG DNA. Okay, so dendritic cells can have toll-like receptor 9, B cells can have toll-like receptor 9, and so on. Okay, but as for epithelial cells, we can focus on the fact that toll-like receptor 5, right, is on epithelial cells, like enterocytes, for example, on the cell surface and can respond to flagellin. Okay, and then of course antigen-presenting cells like dendritic cells have toll-like receptor 4 on the cell surface, which can respond to lipo-oligosaccharides. So toll-like receptor 5, which is present on the cell surface of epithelial cells, can respond to flagellin. Toll-like receptor 4, which is present on cell surface of antigen-presenting cells like dendritic cells, can respond to lipo-oligosaccharides. And then toll-like receptor 9, which is expressed in cells like your B cells and dendritic cells, and it's actually in your endosome, right, your inner lining of your endosomal membrane, and can respond to unmethylated CpG DNA. And Campylobacter jejuni has all of these pattern-associated molecular pattern or pathogen-associated molecular patterns, okay, or PAMPs. But we're going to focus on the lipo-oligosaccharides, okay? So let's say our Campylobacter jejuni, right, due to the contaminated food you ate, goes to your ileum, then infects your epithelial cells. Then from there, it makes its way to the lamina propria, right, which we know is the layer beneath your epithelial cells. In your lamina propria, you have many immune cells, like B cells, like macrophages, like dendritic cells, and so on, right? So you can have a lot of immune cells in your lamina propria, all right? So again, like B cells, T cells, dendritic cells, macrophages, and so on. So let's say this Campylobacter jejuni encounters a macrophage. Your macrophage phagocytoses the Campylobacter, right, jejuni, breaks it apart, and presents it on MHC2. Then it goes through the lymphatic vessels and goes to a nearby mesenteric lymph node, right, draining your intestines. And remember, along with that, you also have free bacterial antigens too, right, of Campylobacter jejuni that makes its way through lymphatic vessels and into your mesenteric lymph node too. Okay, let's keep track of that also. That's important. But so let's see what our macrophage does now, right? So it has its Campylobacter jejuni antigen right here, right? And let's say this antigen is part of lipo-oligosaccharides, right? Which is the pathogen-associated molecular pattern, right? So let's say this is an antigen that's part of the lipo-oligosaccharides that which is one of the pathogen-associated molecular patterns that Campylobacter jejuni has. So once these macrophages make its way, right? Once it makes its way into the mesenteric lymph nodes, it could present this antigen, this lipo-oligosaccharide antigens, to T cell receptors that are all in your lymph nodes, right? We know that, right? So we have T cells in our lymph nodes, a naive CD4 cell, because we know CD4 binds MHC2. So once the interaction happens, we need a second signal too, right? A co-stimulatory event. So your B7, so B7 gets upregulated, right? And expressed on MHC2, because remember, not on MHC2, on the macrophage, right? Because remember, macrophages are activated now, and it interacts with CD28 on the naive CD4 helper T cell. Once this happens, your macrophages can release interleukin 4, for example, and act and which will lead to the differentiation of your CD4 T cells into the Th2 subtype. So what cytokines do the Th2 subtype release, right? It releases interleukin 4, interleukin 5. And remember, interleukin 4 and 5 do what? They help your B cells class switch and produce antibodies such as IgG, IgA, and IgE. Okay? So that's what's going to happen there. So we're going to get antibody production. But first we have to see the B cells, right? Because once you secrete these cytokines, right, we also need the B cells to be activated in order to produce antibodies. So remember, we said there can also be free bacterial antigens coming, and we said we're talking about lipo-oligosaccharide in this example. So let's say we have a lot of bacterial antigens that come in, right, and bind onto B cells that are also in your lymph nodes. Remember, these are IgM, right? IgM or IgD, right? Surface, these are B cell receptors. When your B cells bind these antigens of the Campylobacter jejuni, it can internalize it and then present it onto MHC2. And B cells are also antigen presenting cells. Okay, so let's say it does that. Then T cells, right? CD4 T cells in your lymph node can interact. So this is our CD4 right here, binding MHC2, our T cell receptor binding the Campylobacter jejuni antigen. And then we have, remember, and then when we're talking about B cell and T cell interaction, we need our CD40 ligand on our CD4 T cell binding, what's this? CD40 on our on our B cells. Once this interaction happens, then your T cells get activated. Then your T cells can release interleukin 4, interleukin 5, and then these interleukins can now, right, cause your B cells to differentiate and cause your B cells to class switch, right, into IgG secreting plasma cells, IgE secreting plasma cells, and IgA secreting plasma cells. Okay, so that's how you start making antibodies against this Campylobacter jejuni. Now, normally that would be good, right? But what happens is, right, in people um that get Guillain-Barré syndrome, is you start making antibodies, right? Let's say you start making IgG antibodies against the lipo-oligosaccharide antigen, right? Which is part of the antigen that makes up the Campylobacter jejuni bacteria, right? So that's all good, right? So this will go in and it'll try to bind the pathogen, and your immune system will get activated, complement will get activated, and then you'll try to destroy the pathogen, which is all good. But the thing is, the lipo-oligosaccharide antigen of Campylobacter jejuni has a very similar structure to your ganglioside proteins, of myelin on your peripheral nerves. So not only and that's called molecular mimicry, right? Which is what we're talking about. So the structure of your lipo-oligosaccharide, that's part of that's one of the PAMPs, right, of Campylobacter jejuni. The structure of that mimics the structure of ganglioside proteins on myelin of your peripheral nerves. So that's called molecular mimicry because not only will this IgG antibody attack Campylobacter jejuni, it's also going to attack your ganglioside proteins on your peripheral nerves. Okay, so let's go and see what we have here. So again, right? We make our IgG antibodies, let's say, against the lipooligosaccharides, which is good. That's what we want to do, right? To destroy Campylobacter jejuni. But what happens is the lipooligosaccharides, the structure of that mimics, right, our ganglioside proteins of our myelin and our peripheral nerves. So not only will these antibodies attack lipooligosaccharides of the Campylobacter jejuni, it's also going to attack and bind, I should say, it's also going to bind onto myelin, which will then do what, right? Which will then do what? So once this binds onto myelin, it's going to activate complement, which we know, right? And once the complement system activates, we know that leads to destruction, but it also activates immune cells like macrophages, for example, and neutrophils. And then this will lead to further destruction, right, of our myelin. Okay. So I hope that makes sense now of why we call this molecular mimicry. So now let's just talk about the findings for Guillain-Barré syndrome, right? So let's go from the top. So let's say this is our myelin, right? These are our Schwann shells, our Schwann cells, right, that make the myelin, and we make antibodies not only against lipooligosaccharides, remember, against our ganglioside proteins that make up our myelin. So we're going to see antibodies binding here. We said it's going to attract complement, and the complement system will attract immune cells like macrophages, and that'll lead to the destruction of myelin. Okay, which is called Guillain-Barré syndrome. Now, since we're talking about Guillain-Barré syndrome already, right? We should talk about the different variants. So when we're talking about autoantibodies, right? We can have three main types that we should be familiar with. We can have anti-ganglioside antibodies, like we discussed, specifically anti-GM1 antibodies, right? Which is the which is targets your Schwann cell myelin, like we discussed, right? So, or the normal Guillain-Barré syndrome that we talked about in the previous slide. You can also have anti-GD1A antibodies, anti-GD1A antibodies. What these antibodies target is the axon membrane, okay? The axon, all right? So it targets the axon instead of the myelin. Okay, so this can lead to a rapid, more severe motor loss. Okay, so this can be more severe and lead to a rapid and more severe motor loss. Okay, so that's the anti-GD1A antibodies, and then the other type of antibodies you can get, right, is anti-GQ1B antibodies. Okay, and what these antibodies target, right, is are the cranial nerves. Okay, cranial nerves. It can target the cranial nerves, and it can target your cerebellum. Okay, specifically your cerebellar neurons. So let's see what will happen here. If it targets your cranial nerves, like let's say 3, 4, and 6, that can lead to ophthalmoplegia, right? And if it targets your cerebellum, we know cerebellum is important for coordination, balance, muscle tone, and etc. So if it targets that, we can get ataxia and you can also get issues with your reflexes, right? So what is it called? What's the variant called where you have anti-GQ1B antibodies, right? That target your cranial nerves and your cerebellum. What is this called, right? This is called Miller-Fisher syndrome. Okay, so that's important to keep in mind also. This is called Miller-Fisher syndrome. Okay, where you can get ophthalmoplegia, you can get ataxia, and even issues with your reflexes. Okay, so you can also get areflexia too. All right, so that's that. But if we're talking about normal Guillain-Barré syndrome, right? What can we see, right? What clinical findings can we see in Guillain-Barré syndrome as a whole, right? So we can see ascending paralysis or ascending weakness, right? So usually starts in the bottom part, like your legs, and then it goes and it ascends upwards. And even end stage, right? When it gets really severe, it can also cause paralysis of your diaphragm, okay, which isn't good because you need your diaphragm to expand your lungs and like inflate and deflate your lungs. So, or mostly inflate your lungs, right? And if you can't do that, then that can lead to respiratory failure. So we have ascending paralysis. If it gets really severe, then paralysis of your diaphragm, right? Usually that's like the last stage. And remember, peripheral nerves are important for reflexes. So if your myelin's getting destroyed, right, then you're not going to have proper conduction of action potentials down the nerve. So that's going to affect your deep tendon reflexes. So you can lose your deep tendon reflexes. And of course, if your peripheral nerve, the myelin of your peripheral nerves are getting destroyed, that can lead to peripheral neuropathy, right? That makes sense. And then you may also get bilateral facial palsy if, right, your facial nerves get affected, right? So that's a cranial nerve, right? Your facial nerve. If your facial nerve gets affected, you can get bilateral facial palsy. Okay. So that's for the basic findings. And then that's basically it for this video, right? The main thing was to talk about molecular mimicry, okay? And and I feel like it was good to talk about Guillain-Barré here because we saw a clinical correlation, right, between molecular mimicry and real life, okay, and how molecular mimicry is related to the pathophysiology of Guillain-Barré syndrome, okay? So I hope this video helped. I hope it made a little bit of sense, and I hope it wasn't too confusing. Thank you guys for watching, and see you guys in my future videos.